It's hard times, but let's do this. And let me thank David for the invitation. And it's really an honor. I knew Carl as a student in our computational summer school, Woods Hole, some years ago, and then he came back even to our neuroinformatics sort of data analysis school. So let me just give you the big picture here for a second. I mean, we, as experimentalists, as theorists, we study processing in the brain, dynamics in the brain, computation in the brain. There's another network in the brain, and that's the vascular network, and it serves at least two and maybe three purposes. And one, of course, is to deliver nutriments, deliver oxygen, deliver sugar, deliver neurotransmitters within the bloodstream throughout the brain, make sure that your neurons stay healthy, have their energy supplied, make sure all the other non-neuronal brain cells stay healthy and have their energy supplied. The endothelial cells that form the tube and form the blood vessels themselves, as I'll describe, also conduct signals, just like neurons conduct signals. And while neurons have positive going action potentials, the endothelial cells have hyperpolarizing negative going action potentials that signal the need for increased resources. So the last issue has to do with our removal of so-called interstitial fluids. There's no lymph system per se for disposing of proteins in the brain. They go into the extracellular space, and at some point they come out and on the surface of the brain and the arachnoid space. That's very much a topic in progress, this sort of pumping of interstitial fluids. But all along the way, I think there's a lot of opportunity for theoretical issues that I'll bring up. And I'll also bring up that we got into this because we were trying to measure neuronal activity, and it was confounded due to the movement of red blood cells in the brain. And it somehow, as time went on, we tried to meet the confound, and it just got more and more interesting. So I hope I could share some of this enthusiasm with you. Okay, so let me do this. Okay, needless to say, the wonderful thing about being at a university is that it's extremely difficult to tinker in your own lab because you teach, you write grants, and you often have to deal with, just to paraphrase Woody Allen, those that could do run laboratories and those that can't do become administrators. We've had many fantastic students and many fantastic senior colleagues over the year identify who did what. As we go along, the people in red are people in the lab who are currently working on issues in vasomotor activity. One fellow who's not in this picture, Tomas Bergini, is now a professor in Frankfurt. And what I'm going to do is to give some background. So I'm going to pull work from the past, some of it 15 years old at this point, and then I'll sort of transition to ongoing work, things that are not published yet, but I think are experimentally on solid ground. Okay, so the first thing you need to work out is what the architecture is, or the so-called angioarchitecture, the vasculature in the brain. And to do this, we basically want to make a plumbing diagram of the vasculature. This was done by filling all of the vessels with a gel and having a fluorescent indicator within the gel, and then using a technique just called just the serial two-photon technique. It's an adaptation by a former student, now professor at UCSD, Phil Tsai, of Winfried Dank's two-photon technique. And what you see, this little cube here is about 240 microns on edge, and we filled all the vessels. We have a pretty picture of the vasculature, but we want to turn this pretty picture into an annotated graph. So what annotated graph means is that we want to connect all the vertices by a vessel. We want to know something about the resistance of the vessel. So we want to map the length of the vessel, and we want to map the radius of the vessel. Okay, now solving this, it's very fashionable now to use neural networks for everything, but we're theorists, so we can do things correctly. We know what the point spread function of our light is. That's this little football label, PSF. And we could calculate what the overlap is of this PSF, or more proper convolution of the PSF, with vessels of all diameters and all angles. And from that, we could calculate what we should be measuring in the laboratory. We do that, and that will enable us to actually work backwards now and complete this graph. As a cross-check, we compute the calculated point spread function at the end. It's different in the axial direction than the lateral direction because light is not coming in isotropically into our sample. And so this gives us the graph, and then we could also want to understand what the systematics is in these kind of experiments. In order to do that, we could image the vessels in vivo within sort of the cortical mantle to a limited extent, maybe the first 700 microns or so, as I'll describe later. And then after we perfuse the animal, we know what the diameter of these vessels are. So things work pretty well. They're pretty linear until we get down to the smallest of vessels, which are about 4 microns in diameter, 2 microns in radius. And at that point, the perfused animals have vessels that start to shrink. So we have to correct for this. So the smallest vessels, of course, are going to suffer some systematics. Okay, so what do we get from this? This is actually an earlier picture by Pablo Blinder, who's at University of Tel Aviv, professor, Phil Sy at UCSD. These are the key features that we're going to use in this discussion. These red vessels on the surface are called the Peel arterial network, and this is this branching, and as you'll see in a moment, this interconnected network of vessels that are supplying blood across the surface of the cortical mantle. Okay, and then there are places where there are vessels that plunge straight down into the so-called parenchyma, or the depth here of cortex per se. These are called penetrating arterioles. So we only need to learn 2 words: pia, surface, penetrating, diving down. Okay, and these vessels feed this sea of microvessels or capillaries that are shown here in gray. So the capillaries form a completely uniform connected network across the entire brain. Okay, so this is a completely connected one component graph after it's properly reconstructed. And the other thing is there's no particular rhyme or reason between where these vessels dive. It's an urban legend that you get one vessel per cortical column, or that you could look at the vessels and tell exactly where you are in the brain. It ain't necessarily so. Okay, so our contribution here is that there's this large 3-dimensional subsurface network. It contains about 6 million vessels. You know, it's a big number, but it's very countable. Let's understand what the nature and the functional impact is of this particular vascular geometry. So let's go back to the surface. And this is data from Pablo Blinder, mentioned before, Andy Shih, who's at University of Washington in Seattle. So we're looking just at the very surface vessel. The surface is fed by these great cerebral arteries. This one is fed primarily by the middle cerebral arteries coming up on the side. The key thing is all the vessels are highlighted in green, but the vessels that form loops on the surface here are highlighted in black. And the thing you see immediately is that this is a network, and this has a number of obvious experimental implications. One is that blood could enter from all different directions, and this was a body of work that was pursued with Anna DeVore, now at Boston University, some years ago. The second thing is you might say that this is somewhat insensitive. That was to break an individual loop because there's many pathways. Okay, so this should kind of remind you of traffic in Boston, if you will. Okay, so we introduced a little bit of high technology. We're bumping up on 60 years of lasers in biology, and probably most famously put forth in the James Bond movie Goldfinger. Ours is a little bit of a weaker laser than this one, but we were able to adapt this technology to actually this plus molecules that, when excited with light, would make a free radical that we could use to block flow in order to do a perturbation experiment. And that is measure flow in all the branches of one of these loops as shown here, This is work by Chris Schaefer, now a professor at Cornell. We can map all the flow, and then we could go in and we can make a block at one particular location, and we can see how the flow rearranges itself. So at this point, there's dozens of animals, maybe 100 different vessels. And what happens is that the flow will rearrange itself. One of the downstream vessels always flips in direction, but the magnitude of the flow is essentially unchanged. The system is insensitive, if you will, to damage, okay? So the contribution here is that blood flow to the surface arterial network is robust. It's a single point occlusions consistent with what radiologists report by not seeing block flow on the surface leading to strokes in humans. So let's turn our attention now to flow in this subsurface capillary network. Okay, so we're back again. I showed you this image before. This is a computer-generated image from fitting the vessels with tubes. And I want to look at this fully connected subsurface capillary network and understand it's a little bit of its topology and geometry. So the first thing is this is also a network. And is this network also impervious to perturbation, to single point perturbation? So here we have to use a little bit of a fancier technique. Actually, a few years ago, Donna Strickland, Jerry Moreau got the Nobel Prize for expansion and compression of and amplification of laser beams. We're using their technology. We could maintain a focus below the surface, not damage anything outside the focus, and lead to a similar blockage or a clot. A little too much power. We're back to this James Bond-like picture of exploding a vessel, but with just the right amount of power, we could actually make a block in a single capillary. And this is part of the thesis work of Nozomi Nishimura, also at Cornell University now. So let's see what the consequences of blocking one vessel. So we block one vessel that's shown by this cross, and now we look at the downstream flow. So unlike the surface, if we go one vessel downstream, we have actually a deficit in flow in the single capillary, but by the time we go about 3 branches away, the deficit has disappeared. And again, by doing histology and looking at the viability of the tissue, we find out that this does not lead to any damage. So again, we're insensitive to single point damage. And let's see if we can understand this lack of sensitivity. So Shang Ji in the lab, as part of his thesis project, and it's a collaboration with Jayaman Chandrasekhar, who's now at the Allen, did this full reconstruction of all the vessels within the mouse brain. And so one just topological feature, just to make everybody aware, is that the vessels always meet as triplets, okay? So we could use that knowledge, and we could use our knowledge of lattices and ask if I just had a lattice completely of triplets, right? I mean, in 2 dimensions, a triangular lattice. This is 3 dimensions. What is the percolation threshold? And there's a number, it's about 0.45. Now, we can go in and we could do a numerical experiment because we have the complete vasculature, and I could just start removing capillaries at random. And I could do this numerically at all different structures in the brain. And as I'll describe soon enough, there's about a factor of 4 difference in the density, the length density, number of vessels per volume. And the point is that at first I'm pulling off vessels. I've now reduced, I've taken about 10% of the vessels away, and I still have a fully connected graph. And I take 20% of the vessels away, and more or less I have a fully connected graph, 98%. And the system just fails catastrophically. So this justifies, from a theoretical basis or numerical basis here, the previous finding that knocking out one capillary is not going to affect flow within the brain. Okay, so the contribution here is that the network is initially resilient, the microvascular network, to the removal of branches, and then it fails very rapidly. Okay, so let's go back to the local variations in geometry and see what we learn from that. Okay, so because we have this complete reconstruction, now I could just look at some sort of a statistically average quantity, which is the number of the length of vessels per volume. So it's units of 1/area, millimeter vessels per millimeter cube of brain. So this is a slice, numerical slice, in which we average the density, and all these little names here are just different regions of the brain taken from the Allen Atlas. We just, the exact borders are not terribly important here. We just want to use this information to align it with existing data on metabolism. So what's shown here on the right is the density, okay, of the vascular, the length density for different areas. So places like the inferior colliculus, which are associated with hearing, okay, have a very high density. Okay, that's sort of the last sense, you know, that goes. On the other hand, white matter areas and this yellowish areas, the corpus callosum, have a much lower density. So there's about a factor of 4 variability across the brain. So let's try to use this to our advantage. Okay, so what we've learned is that the vascular topology is uniform, but there's a 4-fold difference in the length density across different areas in the brain. And now I'm going to get back to this issue of the blood supplying glucose and oxygen to within the entire brain, and see if I could relate the density to the metabolic load. So in the 1980s, there was this big movement to use techniques like positron emission spectroscopy, dideoxyglucose, which is a glucose with the last hydroxy group is replaced by an oxygen, which is a radioactive label or some other label. And there's this beautiful experimental study that was done by Hibbard, in which he measured the glucose consumption in all different parts of the brain. Now, we measured the vascular density in all different parts of the brain. So if we take Hibbard's data and our data, we can just plot it, okay? And we get a scatter plot. And these are all these extended data points with our best estimates at the variability. So you can see that there's a nice monotonic relation between increasing consumption and the design of the brain supplying an increasing vascular length density. Now, one can understand this on the average in a simple way. I didn't want to go through the calculation here. This was a calculation by Shangji, again, senior grad student just finishing up in the lab. Because we've measured the vessels everywhere, we can find local regions where you're as far away from a capillary as possible. We find out that these local regions will coincide with a concentration of glucose is going to 0 from calculations. We could put this into a diffusion model. So we know the concentration at the blood vessel. We know that the slope is going to 0 at these places where the concentration reaches a minimum. We also know the concentration there. That's one too many parameters for a diffusion equation, but we could just have an extra phenomenological parameter, which is order 1. And the point is that we could get a very simple, sort of first-year graduate student relationship. And this allows us to make a prediction that the oxygen concentration at the vessel compared to at the minimum falls off by about 16 millimeters of mercury. So this is a prediction that's out there, which is something that we, our colleagues, have to measure and gives you some kind of scale. Okay, so the contribution here is that there's a local variation in metabolic load and that it's monotonic with the length density of the underlying vasculature. It could be modeled in a simple way with a diffusion-like analysis. So now let's turn to this last piece of the vasculature, which are the penetrating vessels. So these sort of one-dimensional vessels. Okay, so this is a numerical slice taken out of the reconstruction data of Pablo and Phil Tsai. And there are these vessels that drop down, these penetrating vessels that supply blood from the surface to the depth. And the question is, how necessary is each penetrating vessel? And I mentioned that the capillaries are fully interconnected. So if you lost the penetrating vessel, in fact, you fill in blood from the side, or is the resistance of this vasculature just too high? So we again go back to our optical technique, and this is a second part of the thesis work of Nozomi Nishimura. Nature was kind to us, or convenient to us, but many of the penetrating vessels, instead of diving straight down from the PO vessels, actually have a little branch on the surface and then dive. That enabled us to just use this surface clotting technique, which is simpler, to block the vessel. We could just chase it with 2-photon microscopy, convinced ourselves that we clotted the entire vessel. This is just a buildup of dye. And what do you find? Okay. What you find is that if you block one vessel, you completely kill off all of the tissue in a tube, which is about 500 microns, okay, in diameter. So this is shown, I think, most nicely by subsequent data by Pat Drew, now a professor at Penn State. So don't worry about the green cells are just called microglia cells. They respond to injury. So this was a convenient preparation. The vessels here are candy colored in red. Before we do anything, you can see a diving vessel go down. It's candy colored in red. We make a block, just like I showed you before with Nozomi. We come back about 4 days later, and the fact that this region is bright red is an indication that all the neurons have died in this region, and the vessels have started to disintegrate. So one vessel, okay, actually kills off this large volume of tissue. We need to understand this. And may I say, you should always wear your bicycle helmet because of these kind of experiments. Okay, so, I mean, the first thing is we have this complete graph, right? So we should be able to predict this effect. So this was analysis done by Phil Tsai. Phil took our reconstructed data and just one by one blocked every penetrating vessel, saw what the effect on flow was. This is linear analysis, okay? This is just the simplest thing you could do. Is it's really just Ohm's law turned into, you know, thousands or millions of resistors, and that gives you this red curve on average, quite a lot of variability. Subsequent to our work, Nozomi, as professor at Cornell, went ahead and remeasured this effect in terms of branching order, which was much more convenient for analysis. And these are these green dots. There's quite a lot of variability, but the important point is that the kind of 50% point for recovery is about 5 vessels down the line, and the point for full recovery is about 9 vessels down the line. So they match very, very well. Okay, so we don't gain any insight from this. This is just a consistency argument that the anatomy and the dynamical measurements are the same. To get a little bit of insight into this, we made some model of the vasculature, and let me just bring up a few features. So the red dots here are feeding arteries. The blue dots are the return veins. The little resistors in between are is the resistance of the capillary bed. These are capillaries in 3 dimensions. So to the extent that you can model these as electrical resistors, what we know is that, you know, in 1 dimension, of course, as you go further and further away, the resistance goes up linearly. In 2 dimensions, if you have a network, it goes up like the log of the distance. And in 3 dimensions, as you go further out, you're picking up more and more parallel circuits, and the resistance goes to a constant. So modeling this as independent resistors was reasonable. And what's happening is that blood is trying to come in from the outside, from these unperturbed vessels, but the resistance of the penetrating veins, as well as, of course, the penetrating arteries, is so low in comparison to the resistance of the capillaries that the blood is just being sopped up by these return pathways. So it's sort of like the veins are acting as a ground plane, if you will, and sopping up the blood and not allowing it to go into these regions. And this simple kind of 2-dimensional lattice model, again, it seems to predict pretty well with absolutely no free parameters what the cutoff is. So that's our idea, and you get a similar prediction for what happens when you remove a vein. This is subsequent work by Nozomi and Chris Schaefer at Cornell. So this is a picture. Now, this the summary from this background is that there's this two-dimensional surface I mentioned. It's fully connected. It's robust. There's this three-dimensional subsurface network of capillaries. It's robust. And the one-dimensional interconnections, being one-dimensional, of course, are extremely fragile. And we have some idea of why nature works the way it is. Okay. And I mean, the impact of this is medical. If you block a penetrating vessel, this could actually cause dementia. And Andy Shi had some beautiful work indicating how blocking one vessel can actually affect decision-making. All right, so this raises, I think, an open theoretical question. I mean, this is the vasculature as it is in the normally grown-up mouse. And the question is, why did nature choose this particular distribution system? You could, think, have a glass of wine, imagine that there may be other potentially more robust systems. There's an experimental challenge to try to actually build them in an animal genetically, but I think it'd be interesting to think about this from a theoretical point of view. All right, so now I want to switch the focus of the talk towards back to the arteries. And the picture I presented is a static picture, but the vessels aren't static. In fact, the supply vessels, the PO arterioles, the penetrating vessels are coated with smooth muscle, and the smooth muscle is oscillating. And we want to understand the effect of this and what the consequence of this is, both on physiology and in terms of measuring techniques like fMRI. Okay, so we're going back. For now, I just want to focus back on the surface. So we're looking at these vessels, the PO vessels on the surface. Okay, and then I'll also talk about the vessels that drop down from these, which are the penetrating arterioles. So there was a lovely experiment done by George Oslo, and back in 1988, this was done at the University of Vermont, which, for those who are curious, is the smooth muscle capital of America. And George is rather heroic. George isolated actually a penetrating arteriole, ligated all the branches, brought it up to physiological pressure, and then started bringing up the temperature. So what's shown on the top trace is the diameter of the vessel as a function of time, and the bottom trace is just the temperature of the solution. And this, when it hits close to physiological temperature, it hits about 35 degrees here, C, it starts to oscillate, and it's oscillating relatively slowly. This is roughly once every 10 seconds, or about 0.1 hertz. So the vessels, the smooth muscle in the vessels, are intrinsic oscillators. So just keep these in mind. This is an intrinsic oscillating system. Okay, so we want to make some kind of measurements. I mean, we and others have known for a long time that that these vessels will oscillate in vivo, and we want to understand what the consequence of this is on blood flow. Now, the veins, the arteries, the whole brain is moving up and down. And so we'd like to do two things. We'd like to measure the diameter of the vessel, and we also want to measure the flux through the vessel. We want to do it precisely. So we go back, we augment the original two-photon method in a couple of ways. One is that the index of red blood cells is very high. So we need to correct for this high index of distortions caused by that. The other thing is that we want to become as insensitive as we can to motion. So just in a nutshell, we make what we refer to in the lab as a curtain of light that envelops the vessel. We do it by extending the beam in a controlled way. It's a clever technique due to Fioca in Texas. And then in order to get a second dimension, extended in a second dimension, we actually scan the beam back and forth at about 16 hertz. So you make this little curtain, and basically a red blood cell moves a small fraction of a micron during that time, and that allows us to target all these individual blood vessels with this little curtain of light and measure flux and measure diameter. So the kind of data that we get is shown here. It's a very short instance of time. The bar here is 1 millisecond. And so the diameter is shown on top. A small fraction of red blood cells are labeled with a different color fluorophore, so we're just counting. And at the end, we get this kind of raw data where you have diameter and you have flux, okay? So, and if I just blow up a little piece of this, you could actually see across time this sort of bulge expansion in a blood vessel. So again, these are effectively point measurements, okay? So what we learned from this is, as I said, the diameter of the vessel is going to change as a function of time. This is going to serve as reference data. The frequency varies a little bit across time. So this data has been warped into phase. So this is phase in the vasomotor cycle. I think the important thing to see is that the diameter change is significant from peak to trough. We're running at around 10 to 15%. Now this serves as a reference to measure the flux. And the flux is, now, you don't really know what the flux measurement is going to be, because if the resistance of the microvessels is extremely high, right, then the vessel is just, it's like a washing machine, just going back and forth, okay? But it's not changing the amount of fluid that's going to move into the brain. But what we see, in fact, is the flux is varying somewhere between like 15 and 20%. So is that a meaningful number, right? These are background fluctuations within the brain. Well, okay, as I said, there's about a 20% change in perfusion in this so-called resting state, non-stimulated state. And let's compare it with a stimulus. So this is data, actually the relevant data here is the data on the left. This is data from Anna DeVore's lab. We're over the part of cortex, which is sensitive to movement of a vibrissa. How much the blood changes due to a sensory stimulation depends on the stimulation parameter, but this gives you a sense of the scale. This is a very short little puff to the vibrissa in the animal, and we're getting a 10% change. And that is on the same order of this background change. So I think this is an important issue, that this ongoing fractional change in the perfusion of the brain, okay, is on the same order of or exceeds the kind of change you get with the stimulation. And this poses an experimental challenge, which is yet to be met, because we don't see changes in cognition, okay, on this 10-second period, okay, but there's yet to be a time-locked experiment of vasomotion to test this. Okay, the other issue here is, what does this tell us about the network resistance? And I kind of hinted at this. So we're measuring the change in diameter and the change in flux simultaneously. So I could, of course, plot one against another, and I could plot the relative change in flux with respect to the relative change in diameter. That's this data here over on the far right. Now, if this was an open pipe, like water coming out of your garden hose, of course, you expect that the flux goes like the fourth power of the diameter. But so the slope here should be 4, but the slope is actually relatively small. The slope is 1.3. So how you interpret this is somewhat model dependent, but this tells us that the bulk of the resistance in the vasculature exists within the microvessels. Okay, so again, this opens up, I think, some important theoretical issues. I mean, people are worried about relationships between current flow and resistance in power networks, but as best I could tell, there's zero work on the relationship to flow and, say, the diameter of the pipes, the resistance of the pipes in vascular networks, or let's just say plumbing networks, okay? And I think the other thing is that what we've really done, we've made a single point measurement, which should, in the context of a model, tell us what the resistance of the capillary bed is. And one, I think from a theory and actually experimental point, one wants to know if we can make maps of the resistance of the microvasculature by making these simultaneous measurements. Okay, so the vessels are oscillating, and we know from fMRI experiments that changes in neuronal activity will influence blood flow, right? We know this from infrared experiments with light. So the question is, how does neuronal activity actually get a handle on this very, very low frequency signal, this 0.1 hertz signal. And just as motivation, I think we're all familiar with the work of Kuramoto. If you have two oscillators and they have some interaction, that if their frequencies get close enough, they could actually lock. So whether it's two oscillators with a symmetric interaction, if the interaction is strong enough, they'll lock, or even if it's one oscillator pumping another, if their interaction is strong enough, they'll lock with just a very slightly, with a factor of 2 difference in this little equation. Okay, so the data to understand what the neuronal signal is actually comes from human data. This is an experiment by analysis by Rafi Malik at the Weizmann Institute. This is based on human data taken by Fried. This is an unfortunate case of epilepsy patients that had many electrodes. As the surgeon, Yitzhak Fried, tried to find out where the locus of the epileptic center was. These are rough measurements. I mean, they're done in a clinical setting. Nonetheless, if one takes this time series of surface measurements and just looks at its power spectrum, it's a very, very bland power spectrum. There's nothing to see. But if you chop up this power spectrum into small intervals, and here I'm chopping them into 2 10-second intervals shown here in red and in cyan, you can see that the low frequency signals are the same, but the high frequency signals have changed. And then to do this in a more purposeful way, we take this time series, we compute its spectrogram. And if we look in this relatively high frequency range of here, it's 40 to 90 hertz, right? What you can see is that the power, okay, is just waxing and waning. So that the envelope is waxing and waning. And I could just strip out the envelope as shown here, band limited local field potential power. And you can see that this has fluctuations on this kind of order 10-second time scale. And if I just take this power spectrum of this, the so-called second spectrum, you can see there's peaks at around 0.05 and around 0.15 hertz. So basically, what this is saying is that there's no process that we know of in cells that generates this 0.1 hertz, but for reasons yet to be understood, there's an envelope over the high frequency signal. So the lesson here is that the so-called gamma rhythm, high frequency signal, which all of you hopefully are producing if you're attending to the talk, is the source of this ultra-flow neuronal activity. And this led to the hypothesis that we have this band-limited, envelope of the gamma band power that's influencing the diameter of the arterioles. So this was tested in the laboratory by Salim Mateo. Conceptually, it's a relatively simple experiment. This is a rodent. For technical reasons, you don't want to remove the skull. I make an open volume system. You make a thin skull. You measure the diameter of the vessel, again with 2-photon imaging. At the same time, we're recording the electrical activity of the brain within this region. I'm showing here the spectrum of the local field potential, and you can see there's bursts in activity within this region. If I extract out, like I did in the data from Rafi Malik, the band-limited gamma band power, okay, that's shown as the gray trace. If I just look at the diameter of the vessel, that's shown as the magenta trace. You really don't need a theorist to see that these things are pretty correlated. And if I just compute the normalized correlation function, they're correlated with around 0.5, okay? And the square of this is roughly the variance explained. So it's around 0.2, 0.3. And so what we learned from this is that the 0.1 envelope of the gamma band can entrain, partially entrain, right? Peel vasomotion. And this has been published now. We could push this a little further, and it's actually signaling through the corpus callosum. These are layer 5B neurons between two sides of the brain, which allows, say, symmetric regions, symmetric with respect to the midline of the brain, to be synchronous, that you're transmitting this envelope. And of course, this is what you pick up with fMRI, either cerebral blood volume fMRI or blood oxygen level determined fMRI. Okay, I want to give a little bit of background. As I mentioned at the beginning, this is work by Mark Nelson, and I think it's important to understand for how the neurons actually communicate this signal. Mark is consistent with my statement about University of Vermont, at the University of Vermont, and he published what I thought was a gorgeous paper back in 2017. So let me take you through it. What Mark did is he looked at the diameter of a vessel on the surface of the brain, which is here, a penetrating vessel. It's this guy in green, okay? And then he puffed potassium at capillary, about 100 microns or 150 microns below. He puffed about 10 millimolar of potassium. That's pushing the limit. There is not a good measurement of exactly what the potassium concentration is at this small space. So nonetheless, you could see that the vessel got big at the same time that the capillary hyperpolarized, okay? And if you look at the capillary diameter, there's no change, consistent with no smooth muscle in the capillary. But if you looked at the peel artery above on the surface, you could see that it, in fact, dilated. So how do we understand this effect? And this is the picture that emerges. So let's just go through these numbers: 1, 2, 3, 4, 5, 6. The neuron is spiking. The neuron has processes in the vicinity of a blood vessel. And as it spikes, extracellular potassium is building up. Exactly how much depends on anatomical details. As this potassium builds up, there's a channel called the potassium inward rectifier. It's different than the sodium channel in the sense that its activation potential depends on the concentration of nearby potassium. So as potassium is building up, you need a lesser hyperpolarizing potential or more depolarizing potential to turn it on. So as potassium builds up, this channel snaps on. It's a potassium channel. It pulls the membrane potential down. Okay. So this sends a hyperpolarizing signal through the capillaries into the penetrating arterioles and back up to the surface at the peel vessels. Smooth muscle is on top of the peel vessels, and the smooth muscle will now dilate. And this was shown by earlier work from Mark Nelson, where you can look at the diameter of the vessel as a function of potential. And as you hyperpolarize the blood vessel, you get a larger diameter. Okay, so this is what I call the other signaling mechanism within the brain. Okay, so this, I think, opens up a number of theoretical issues. I mean, starting with Rawl, we know how to take the cable equation, right? I mean, from Helmholtz and solve it, and we could put active channels on it, and to some extent, we could not solve it exactly, but we could solve it. But again, as best I could tell, there's never been a single paper on a lattice, okay, of cables and the communication within that lattice and the resistance within that lattice. So I think there's a need for making a lattice of cables in 2D and in 3D, finding out what the asymptotic resistance is and understanding both passive and active propagation, because these signals that cause dilation of vessels eventually die off. And the best we could do is we have some idea from cables, what the attenuation lengths for one-dimensional cables, but how this parses out in 2 and 3 dimensions hasn't been worked out. Okay, we only know about resistor networks. Okay, and like I said, we'd like to know about how attenuation scales with length. And of course, again, just to bring up something else that we think about or used to think about in neurons, these active pulses are colliding. Okay, one has to think about if that's going to cause them to cancel out. All right, so I view this as an interesting theoretical issue. All right, so we know that vessels oscillate. We know that neurons can entrain them. And what's some of the consequence in shaping the the dynamics of the pia. And this is something we measure in the laboratory. We're doing it optically, and this is something, as I'll argue in a moment, has to have a direct effect on fMRI, something I hope to work out with Xin Yu at the Martinos Imaging Center. So we all know about resonance. It's a very general phenomena. You could have just a resistor, inductor, capacitor circuit, and you get a resonance at close to 1/LC. You could have an active oscillator. This is just data. This is just a calculation for a Van der Pol oscillator. You get a resonance. It's not until you get off resonance that you get differences in behavior and can tell whether it's active or passive. So we want to really know if these oscillators, these smooth muscles, are behaving in this simple picture, the simple resonance picture. And we're going to do it by trying to drive them with two different inputs. And these inputs can have frequencies that are close to or far away from their resonant frequency. So let me take you through this, okay? So now I want to look at many vessels. And to look at many vessels at once, this two-photon technique is not so good because it's a scanning technique. And I want something where I could take a rapid snapshots and look at the whole area. So the idea here is to, as vessels, as the smooth muscle begins to depolarize or hyperpolarize, the concentration of calcium in the smooth muscle is changing. So we have an animal that was made by Mike Kolikoff at Cornell, in which the smooth muscle contains an indicator of calcium. This is 8 generations after Roger Chen's cyclically permuted green fluorescent protein. We have another marker at a different wavelength, way off in the red in neurons. That's not going to be terribly important, but using this indicator, I could take snapshots and I could get a measure of the activity, okay, the calcium concentration in these PO vessels. Now, the question is, how good is this compared to measuring the diameter directly? So Tomas Bergini, again, professor at Frankfurt, Jacob Duckworth, grad student at UCSD, performed somewhat heroic calibration measurements where we simultaneously measured the diameter of the vessel, putting a dye in the vessel shown in green, and measured the calcium concentration, which is shown here in magenta. Yes, these are simultaneous measurements, two-photon measurements. And what we find out is that the calcium and the diameter have about an 80% correlation. So we take a little bit of a hit from a direct measurement, but we can measure from lots of vessels at the same time. And we get a little bit of a delay consistent with a multiplicity of past data. Okay, so the basic experiment is this. Let's pay attention to what's happening on the left-hand side, and let's pay attention to the lower left-hand corner. This is a case where all the PO vessels are oscillating at their vasomotor frequency. In this particular case, it's running close to about 0.05 hertz. They're all effectively synchronous. Now, what happens is we're coming in with two different drives. One is a photic drive, we're literally flashing light solely to the eye of the animal at a certain frequency. Here it's 0.0, it's written here. Okay, it's 0.05 hertz. Sorry, it's 0.08 hertz. And we're also driving the vibrissa at a somewhat higher frequency. And we can see there's a region of vessels that are primarily locked to the photic stimulus. And the color here reflects the dominant frequency, okay, that these vessels are oscillating at. So the photic frequency has this area of vessels, and the vibrissa has taken over all the vessels, and that's shown here. Okay, now I know what these frequencies are. In this case, the photic frequency is closer to the vasomotor frequency than the vibration frequency is. So let's exploit this now to understand resonance, if it exists. Okay, so this is the previous case where the photonic frequency, the flashing to the eye, is close to the vasomotor frequency compared to the vibrissa frequency, further away. And we could see how many here. We could just look at the total area of vessels, okay, that are locked to the photic frequency. Now, I flipped the simulation paradigm. And so now I'm moving to this center trace on the bottom. And in this case, the vibrissor frequency is much closer to the vasomotor frequency than the photonic frequency. And here, if I look, I see a smaller number of vessels, okay, locked to the photonic frequency. So this experiment was done on many animals and many vessels. And what's plotting here is the fraction of arterial area, which is dominated by the photonic frequency. And if I'm at resonance, I get the largest possible area. And if my frequency drops below the vasomotor frequency, the natural oscillating frequency of the PO vessels, then the area drops off. And if it's above the natural frequency, the area drops off. So these guys show a clear resonance. So this demonstrates, it doesn't say whether the oscillators are active or passive, but it does say that resonance exists. I think this gives a strong warning, perhaps, to the fMRI community. So our contribution here is that the spatial extent of phase locking among PO vessels depends on both the underlying neuronal input and the local vasomotor frequency. And we're pushing very hard to see if this leads to this kind of parcellation, which has been brought up by the fMRI community in finding different areas of the brain, because we would argue that the size of these regions depends on the stimulation. Okay, so the last question here has to do whether these vessels, we could show that these vessels are really active in the brain. And okay, so George Osso's experiment says that they're in isolation, they're active. What happens within the animal? And part of this is a reaction, I would say, to the fMRI community writing things down like the hemodynamic response function, which basically implies a passive response. So we're motivated in our experiment. There's a number of ways to look for activity. In our case, we're motivated by a comment by Norbert Wiener, and was sort of put on hard ground by Yoshino Kuramoto in his wonderful little book in '84. And the idea is this: suppose you have a distribution of oscillators, as just shown in this little cartoon in the lower left-hand corner, and then I come in with some external drive. Then the oscillators that are close in frequency that the external drive will lock, okay? And their power will add to the power that I supplied by this external drive. And then, of course, there'll be oscillators that are further away in frequency and can't lock. So in a sense, I'll be burning a hole, if you will. This really is like hole burning in spectroscopy. Okay, so this experiment was done by Tomas Bergini, Jacob Duckworth, Karishma Chiabata in the lab did similar experiments. I'm going to discuss Tomas's experiments. We're stimulating the vibrissor region. So this little paw print is the cortical columns within the vibrissor region. And they're just going to be integrating the power from changes in the diameter of all the vessels in this region. So let me take you through this. So let's look on the upper right-hand side. This gray curve is the background power. Okay, so it's falling off with frequency. This is spectral power, and this is log scale. And you can see very nicely, there's a little bump here at about 0.13 hertz. That's the vasomotor frequency in this animal. Now I come in with a stimulus, and the stimulus is just a puff to the vibrissor. So if the stimulus, as shown here, which is coming in at around 0.08 hertz, about once every 12 seconds, I'm going puff, puff, okay? And what you can see is that the background power has decreased. Okay, I'm burning a little hole. And if I remove the applied power, I'm extracting this much power, this given by the extent of this arrow from the system. When you puff, of course, you're not just getting the fundamental, but you're getting all the harmonics. So at the second harmonic, I also extract power. But by the time I get up to the third, fourth, fifth, etc., I'm not extracting any power. So the method I'm using here is a so-called line spectral method. Okay, so we do this, oops, sorry. We do this for, again, very many animals. And there's about 13 mice in here, a couple hundred measurements. And what we see is that between close to zero frequency and up to about 0.2 hertz, which is about the highest frequency that smooth muscle can follow, we can extract power from the system. And the point of this exercise is that these vessels are active oscillators, okay, reacting to their input. So our contribution here is that in line with the prior ex vivo data, brain arterioles are active oscillators, and they are not passive low-pass filters. I just emphasize this. So the summary here for the second part is that the spatial pattern of vasomotion in our picture emerges from a kind of competition. You have neighboring oscillators that are trying to lock each other. Okay, so they're trying to be at the same frequency. I didn't talk about it, but there's some slight phase shifts and therefore some waves that go along in the vessels. And what's competing with these oscillator-oscillator interactions that are acting on the intrinsic oscillators is that there's input from below, and this input from below is also trying to lock these oscillators. So I think of it a little bit like a magnetic system in a disk drive. You have spins, they're next to each other, they're all trying to line each other up. And then you come in with a right head and you have an external field, and that external field is trying to pull it. And with that, okay, let me thank everybody for listening in on a different than the usual topic, and thank David again for the invitation.